Hello everyone and welcome back to Neuropsychology. So in the last video we talked about chromosomes and DNA and genes, amino acids, and the most important things we talked about transcription and translation. Okay, so in this video we're going to talk about why these proteins are so important. So why do we cover how proteins are made and how do our cells use these proteins? So the proteins are embedded into our membranes as channels, gates, or pumps. So we will go over that. Then we will go over the concentration gradient and voltage gradients. And lastly, we will go over how the movement of ions across our membrane creates an electrical charge. So let's get started. Okay, so proteins are so important for neurons to talk to one another. So again, I already said that, but proteins can be embedded in our cell membrane and they can form channels, gates, or pumps. So I said before that nothing goes through the membrane, right? Okay, so technically that's true. However, because these proteins are embedded into the membrane, they can let certain molecules or ions through. So let's start off with channels. So some membrane proteins are shaped to form channels. So channels technically are big holes or some holes through which certain substances can always pass. So they can go in and they can go out of the cell through these channels, but only certain things can go through it. So different proteins with different sized channels allow different substances to enter and leave. So it depends on what protein, on what it lets through. Okay, so next up we have the gates, the protein gates. So some proteins, such as these gates, are molecules that have the ability to change shape. So they can change shape whenever a specific chemical binds to them. So it's kind of like how it works with a lock. So it's a lock that can open whenever a certain specific chemical is attaching to them. So the chemical could be a key, and then they can open up. So technically, gates are proteins that open and close depending on if a chemical is attached to them. And sometimes it doesn't even have to be a chemical, but it can be an environmental change, such as a change in electrical charge or temperature. Okay, so we talked about channels and gates. The last one up are pumps. So pumps also can change their shape. So they're shape-changing proteins, and we often call these transporter proteins. So how it works is they actively transport substances in or outside of the cell. So the only difference with pumps versus gates and channels is that pumps cost a lot of energy. So they take a lot of energy or ATP from the cell. So a really important example is what we call a sodium-potassium pump. So this will pump sodium in one direction and it will pump the potassium in the other direction. So let's go ahead and look at an image to make this a little bit clearer. Okay, so here you see the three different kinds. On the left you see the channel, in the middle you see a gated channel or a gate, and C is a pump. So a channel, it's always open. It can let things in or out whenever it wants. It's very specific though. So in this case, it's a potassium channel. So ions can cross a cell membrane through the appropriately shaped channel. So this potassium particle can go in or out of the cell whenever it wants. Then the gated channels, these are the ones that can change shape. So it can either be closed or it can be open. So a gated channel changes shape to allow the passage of substance whenever it's open. So if it's open, the sodium can go in. And it also prevents the passage when one or both gates are closed. So now one is closed, but they can also both close. And then lastly, we have the pumps. As I said, the sodium-potassium pump is a really important one. So a pump transporter changes shape to carry substances across a cell membrane. So in this case, it will send the sodium out or in and the potassium out or in. So same thing here, it can go out or in. So kind of whenever the sodium goes in, the potassium will go out, and the other way around. Okay, so all the molecules in our body, they constantly move. So whenever there's a different amount of substance in one liquid compared to a liquid next to it, there will be a concentration gradient. So molecules kind of always spread out for where they're more concentrated towards where they're less concentrated. So this is what we call diffusion. So technically, diffusion is the thermal motion which causes molecules to spontaneously spread out from an area of high concentration to an area of low concentration. So here I picked a picture from the internet. If you put, for example, ink in water, in the beginning it's concentrated and then it will move to where there is a lower concentration until it reaches an equilibrium. Okay, so with ions, it's a little bit different. Yes, there's also a concentration gradient, so we have to keep that in mind. But when salts dissolve in water, the ions that are in the water, they have a certain charge, so they can either be negative or positive. And when they diffuse, they do not only go by the concentration gradient, but they also follow their voltage gradient. Okay, so what is this, right? So a voltage gradient is the difference in charge between two regions that allows for measuring the relative concentration of positive and negative electrical charges. Okay, so let's start off with what we call these positive and negative ions. So positively charged ions are always called cations, and I found this picture on the internet. If you see this picture, you will not forget that a cation is positive. It has a positive charge, haha. And then anions are negatively charged ions. So examples of cations are sodium and potassium, and an anion could be chloride, or there are also these little protein anions in our cells which are also negatively charged. So ions always move from areas with high charge to areas of low charge until there's an equilibrium. So whenever it's very positive, they go somewhere where it's less positive. Okay, so let's go over this in pictures. So on the left side, you have an example of a concentration gradient, and on the right side, you have an example of a voltage gradient. So let's go through this one at a time. So for example, when you drop a little bit of ink in water, in the beginning right here, it's very dense and very concentrated. So then eventually, over time, it will diffuse and it will spread all over the water until it is in equilibrium. So on the right side, if you have a salt or salt water, or just as table salt, and you put that in water, salt consists of ions. It has a positive sodium and it has a negative chloride. So if you put that in water, the positive and negative ions will flow down their gradients until the positive charge are equally distributed everywhere, and also until the negative charges are distributed equally everywhere. So as you can see in this picture, if you pour it, in the beginning it's going to fall right here, and eventually it's going to spread out similarly as this one. However, how does that work when there is a membrane in between? So we're just going to be talking about ions in this picture. So on the left side, you have an impermeable membrane, which is a membrane that does not let anything through ever. And then on the right side, you have a semi-permeable membrane that only allows certain things to go through. So let's start with the left picture. So if you place a salt on the left side of the impermeable membrane, it will go down the voltage gradient until the positive and negative ions are distributed evenly. But as you can see, it will not cross this membrane. So it will distribute here, but it will not be able to move over here. So on the right, you see a semi-permeable membrane with chloride channels. So these chloride channels will let chloride through, but not the positive sodium. So if you put the salt, which consists of sodium and chloride, on one side of this semi-permeable membrane, chloride ions will diffuse from the side of high concentration through these little channels to the other side, where it's of low concentration. So if there were no if there was no charge involved, the chloride ions would be half on the right side and half on the left side. However, because there is a voltage gradient, the chloride ions will be pulled back towards the left, where all these positive sodium ions are. So at equilibrium, one half of the container is more positive, which is this one right here, and then the other half will be more negative. So this is actually a really good example of what our membrane looks like of a neuron at rest. So we're going to talk about this in more detail in the next video, but I just wanted to go over the basics before we dive into all that. Okay, so when you think about a membrane, I want you to keep all these things in mind, otherwise it's going to get very confusing. So a membrane is impermeable to salty solutions, and it uses these proteins that we talked about for the last couple of videos to transport substances in and out of the cell. So there's both a concentration gradient and a voltage gradient across the membrane. So we definitely need to remember that both of those two are happening. And that was the end of the video. I will see you all back soon.